Rahim, welcome back guys. Welcome to another video lecture by Kami Biology. Today in this video lecture we are going to talk about MALT or mucous membrane. In the previous video lecture, if you guys remember, we talked the intact skin, which are actually salt and which take a part in the innate immune system and which are the kind of physical barrier. So today what we are going to talk about, mucous membrane or MALT. So let me write. Now, what are the mucous membrane? First of all, let me explain the mucous membrane, and then after that we will explain MALT. Now, what are the mucous membrane? Mucous membrane are those membrane which can secrete the mucus. Okay, we have different part of our body which are specially responsible to produce the mucus. We have our respiratory tract. So let me write in a short form. We have a respiratory tract, urinary tract, digestive tract, and eye. Okay, so these are the part of our body, and such part of our body are specially responsible to produce the mucus. Now mucus, when they produce the mucus, so mucus have antimicrobial property. Okay, they can kill the microorganisms. They produce mucus. Now this mucus have antimicrobial properties. They can trap the microorganisms and kill them. But what actually happen? This mucous membrane, and we have some special cells which we call it stratified squamous epithelium. So let me write. We have stratified squamous epithelium. Now these two, mucous membrane and this squamous epithelium cells, when both are combined together, then both have the antimicrobial property. These two cells can trap the microorganisms. So what they actually do, they will trap the microorganisms. Now once they trap the microorganisms, after that they will cause the destruction of the microorganism, means they can kill the microorganism because they have the antimicrobial properties. Okay, these part of our body can trap the microorganisms and not allow, or they can prevent the microorganisms from the penetration to the sticky area of our body, because microorganisms usually they usually need the sticky area inside our body for their surviving. Now we have different kinds of the mucuses which have toxic property and which are actually toxic against the bacteria, and these mucus are known as cervix mucus. So let me write. We have prostatic fluid, and tear. Okay, so all these are actually the mucuses, and all these mucuses are what? All these mucus are usually toxic to bacteria, means they can kill the bacteria. Okay, moreover, among them there is a lysozyme which contain an enzyme which we called it Murumidase. This enzyme play an important role, which are actually produced or which are specially present in the tear. So let me write. This tear contain what? They contain lysozyme. Now this lysozyme contain Murumidase enzyme. So let me write. So this what are the function of this Murumidase enzyme? This Murumidase enzyme can break the beta 1,4 bond. They are usually responsible to break beta 1,4 glycosidic bond. So they can break this one bond. These bonds are present between the N-acetyl muramic acid and N-acetyl glucosamine. So let me write. What? These are actually the building blocks. If you remember, these are the building blocks of peptidoglycan, means the cell wall of the gram-positive bacteria. So these are the peptidoglycan building blocks. Now this peptidoglycan are actually the cell wall of the gram-positive bacteria. Now how this beta 1,4 bond are present between this neg and name? Okay, we usually call, we also call that neg and name. So how the bond are present between them? So if I draw the structure of name and neg, so it will be look like in this form. These corner are presenting the carbon. Okay, this is the carbon number 1, carbon 2, sorry, the carbon number 1 are start from this point. Carbon number 1, 2, 3, 4, 5 and then 6. Okay, so and again 1, 2, 3, 4, 5 and 6. So how the bond is formed? Now then the carbon number 1 of name, this is the carbon number 1. So the carbon number 1 of name are usually make a bond with the carbon number 4 of the neg. So this is the carbon number 4. So both are connected together and they make a bond. Now this bond is called what? Beta 1,4 glycosidic bond. Okay, and usually it is break down by this enzyme, Murumidase enzymes. Okay, and after that, if the, because all of you better know, if you remember, cell wall are very important for the bacterial protection. So once the cell wall are break down, means these are actually the main these are the main part of the cell wall. These are building blocks of the cell wall. They can make the cell wall. So the bond between each bricks are become break. So then there will be no bond and there will be no attachment. After some time, the cell wall will be a very weak cell wall are formed and the cell wall will be disturbed and destroyed, and finally the bacteria will be killed. Moreover, we have some we have some proteins which we call that specific amino proteins. Okay, so let me write. Now what are the function of each specific amino proteins? These proteins are actually present in the mucus, in secreted mucus, and each protein are responsible to prevent the bacteria from the attachment to the sticky area of our body. Or they don't allow the bacteria to attach on the sticky area. Moreover, this mucus also contain iron binding protein which we call that lactoferrin. Now what are the function of this lactoferrin? These lactoferrin are usually released, or these lactoferrin are obtained from the cells which we call that activated macrophages and polymorphonuclear leukocytes. So now what are the function of each lactoferrin? Once the lactoferrins are produced from the mucus, so what actually happen? This lactoferrin are fall on a bacterial cell. So let's say these are the lactoferrin. So this lactoferrin are fall on a bacterial cell. So let's say this is a bacterial cell. So here these are, let's say these are the plasma. So this plasma contain iron, Iron amount are reduced. So the iron amount are what? Reduce. So how it is reduced? It is reduced by this lactoferrin. So once the iron is reduced by lactoferrin, so this bacteria will be no more longer to multiply. Okay, and furthermore, these mucous membrane can also secrete, if we talked about this mucus, so this mucous membrane can also secrete another important toxic material which are against the microorganism. We call that lactoperoxidase. Now this lactoperoxidase can catalyze the superoxide radical. Now this superoxide radical are actually a reactive oxygen intermediate. These are reactive oxygen intermediate, and after that these reactive oxygen intermediate are toxic to many microorganisms, so they can also kill the microorganisms. So that's all about the main function of the mucous membrane, and that's all about the main features and characteristics of the mucous membranes. Okay, so now let me explain the MALT in detail, and let me erase this one portion. Okay, now let's explain the MALT. MALT, just like in intact skin, if you remember, intact skin contains salt, skin associated lymphoid tissue. So similarly, the mucous membrane also contain a specialized tissue which we usually call that MALT. Okay, so MALT have two main types. So let me write. This MALT have two main types which we usually call that GALT and BALT. So let me write. Now this BALT stand for bronchial associated lymphoid tissue. Inshallah will be discussed in the next video lecture. And they also contain means gut associated lymphoid tissue. Now this gut associated lymphoid tissue, now where this GALT are present, it is usually found in the, let me write, so it is usually found in the adenoid tonsil diffused lymphoid area along with gut. Okay, and they also found in a specialized area of the intestine which we usually call a Pear's patches. Now if we discuss the function of this MALT, so let me draw a simple diagram of the gut. Okay, so I'm not drawing the whole structure of the gut. So let's say if these are the cells, so let's say these are the cells, okay, which are connected to each other, and these cells are usually the cells of the gut. Okay, and with each of the cells there is a layer present outside of each of the cells. So there is a layer. Let me draw as you are watching. These are the layer. Now this layer is called what? This layer is called mucous membrane. Now these cells, as you are watching, these are actually the gut cells. We call that M cells. We also call that what? We actually call that M cells. Okay. Now within each M cells there are the cluster of cells which we usually call it lymphoid follicle. So let me draw each cluster of the cells. Among each lymphoid follicles, they contain B cells. Now what actually happened? If we discuss this M cell, so let me draw a large diagram of this M cells for your understanding. Okay. Near each M cells there are the columnar epithelial cells. Okay, now the columnar epithelial cells are connected. If we let me draw here, let's say these are the columnar epithelial cells. Now let me write, these are the columnar epithelial cells. These columnar epithelial cells contain a hairs like structure as you are watching. Now this is called brush border or microvilli. Now brush border and microvilli are specially responsible to trap the microorganisms and finally they can kill it. Okay, but this M cell doesn't contain the brush border or microvilli. So instead each microvilli they contain macrophages, T cell and B cells. Now what actually happen? If the microorganism contact with these cells, it is very large cells. Okay, so let's say these are the antigen, or let's say these are the any kind of microorganisms. If these microorganisms are connected or attached to this M cells, so what actually happen? This M cells can endocytose it. Now once it is endocytosed, so let's say these are the antigen. So here I will draw just one antigen. Now after that what it will happen? The macrophages are activated and they can engulf it. So let's say these are the macrophages. Now this macrophages engulf it. After that, once it is engulfed, so let's say it is engulfed. Now once this antigen are engulfed, so this antigen are then break down into small fragments, and after sometimes they present this kinds of fragments to their receptor, which we call that MHC2 receptor. Now they can present each fragments to the helper T cells. So if you remember, I talked that here the T cells are also present. So especially here the helper T cells are present. So after that the helper T cells are attracted. So once the helper T cells are attracted to that MHC2 molecules, and once they recognize the fragments of the antigen which is presented by this macrophages, because macrophages are actually the antigen presenting cells. Now after that what actually happen? They secrete the signals to the B cells. Okay, so the B cells which are present among the lymphoid follicles will be attracted to that specific area. So this is the B cells. So once these B cells are attracted, they are converted into a mature cells which we call that plasma cells. Now plasma cells are again antibody producing factory. Okay, now these plasma cells usually secrete mucus associated antibody, which we call it secreted antibody. Secreted antibody. Now such secreted antibody are released to the lumen of the gut. Now after some time, let's say this is the lumen of the gut, or this area is the lumen of the gut. Okay, so after some times, once it is released out, so then it will be attached to each antigen as you are watching. Now after some time, what actually happen? This antibody have the ability to kill the each of the antigen, or sometimes they will attract some other cells, neutrophils, monocyte, or the macrophages or dendritic cell, to engulf each of the antigen. Okay, and finally the process are under the control. Okay, so that's in a sense about the mucous membrane, or that's in a sense about the function of the MALT. I hope you understand about this video lecture. Inshallah, in the next video lecture we are going to talk about BALT and GALT in a detail, so please stay tuned and continue to watch the video. Thank you so much.